All right, guys, so we're going to move on to our next lesson, which is going to be all about the regulation of the cell cycle. And so I decided to go ahead and split it up into two parts because honestly, it can be kind of lengthy and it can get really hard to understand if we try to cover it all in one day. And so at this point, hopefully, after several days with the cell cycle, you feel pretty comfortable with the processes that occur as a cell grows and divides into two new cells. And so the thing is that cells don't just divide because they want to, okay? The process of the cell cycle is highly regulated. And so because of that, not all cells are doing the same thing at the same time for the same reasons, okay? And so it's going to be different for different types of cells in your body. It really depends on what type they are and what their function is. And so we've already talked about how some cells, like nerve and muscle, they're pretty complex in their networks and how they function together as a group and how they are built in terms of their structure and connected to other cells. And so cells like nerve and muscle, they're not constantly growing and dividing. They're in that G0 stage where pretty much they're just being a functional cell, but they're not going through interphase mitosis cytokinesis. And these are the ones that we can't just easily repair because they don't just constantly divide. Whereas certain cells like skin, bone marrow, the linings of our organs like digestive system, they are constantly growing and dividing, growing and dividing, growing and dividing. In fact, we're replacing those linings and those coverings pretty much all the time. And so it really does just depend on what type of cell it is. And so this lesson is going to be all about the different regulators that kind of control the timing of the cell cycle or telling cells to divide. And so the signals can come from within the cell. They could be internal signals. And these are primarily going to pretty much ensure that those stages are progressing and moving in a normal sequence. And they're going to be checking for certain criteria before the cell will continue into the next stage. And so most of these are going to be internal regulators, but we could also have some external regulation where chemicals are coming from outside of the cell and telling the cell what to do. So we're going to start with these internal regulators, and there's two major components, and they really go together. This is a picture over here is really way more complicated than what you have to know, but there's a combination of checkpoints, which are going to be critical stages where certain events are checked before the cell will continue. And they kind of get these stop and go signals. And then what kind of determines what happens at those checkpoints are some proteins. There are different protein regulators. And guys, this is way beyond what we're going to go into. We're going to keep this simple when we talk about these proteins. But regulation and cell communication is a very complex thing in cells. And so we're going to keep it kind of basic here. And so there are three major checkpoints throughout the cell cycle where we get these stop or go signals. And it's kind of like if you were going through school and we have these progress reports and we have report cards. These checkpoints are kind of the same thing. So at a progress report check, maybe your grade isn't doing so hot, and so maybe the teacher decides if you need to come in for extra tutoring or something like that. And so based on what the conditions are at that particular moment, we'll either say move forward or we'll say hold up, we need to take a look at something before we proceed. And that's the purpose of these checkpoints. So there's three different locations. We have G1, G2, and M. And so hopefully we know our cell cycle at this point. And so G1 was our first growth phase. And what was going on is the cell was getting bigger, it was doing normal cell activities, and it was making more organelles. And the G1 checkpoint for most cells is probably considered the most important one. This is the main one that's going to say move forward or stop. And what we're looking for are favorable conditions. Do we have plenty of stuff? Okay, do we have plenty of organelles? Is the chemical environment good? We're looking for the building blocks for DNA, because remember that right after G1 we have the S phase, and we're going to have to double our DNA, so we have to make sure we have plenty of building blocks, which are going to be those nucleotides that we need. And then we're going to check the condition of the DNA itself. And this right here is why this one is so important, because we do not want to copy cells with bad DNA. Okay, and we'll see why at the end of our lesson, because we're going to talk about cancer. And essentially, a bad cell that has mutated DNA is going to possibly lead to the copying of bad cells over and over and over again. And we really don't want to make copies of bad cells, okay? So this is going to be one of our most important ones. If the DNA is damaged, there's a very good chance that the cell is going to receive a stop signal, and it will halt before it continues on to kind of check some things. Now there's going to be another checkpoint at G2. So this one is after the S phase, after the DNA has been copied. So once again, we're kind of looking for proper DNA replication. Do we have the right amount? Did we have any issues with our DNA? And then the third checkpoint is going to happen during the M phase. And so remember, the M phase is where we have mitosis and cytokinesis. And this is specifically going to kind of check around metaphase. We're going to be looking for the spindle assembly. Did it attach to the chromosomes properly? Are they aligned properly? Because if not, those chromosomes may not be distributed properly. And so if it looks like those processes are not correct, then we may get a stop signal here. And so these are just a couple of graphics that show you, okay, so here we have the G1, all right, and so it's at the G1 where if we did have bad DNA or there was something wrong, the cell might possibly enter that G0 stage, as we see here. The G2, we're looking for proper replication, and the M, we're looking for proper chromosomes and spindle attachment. Same picture over here. G1, we're looking for a favorable environment. G2, we're looking for damaged DNA or properly replicated DNA. And over here at the M, we're looking for chromosomes attached to the spindle, ready to be pulled apart. And so these checkpoints are regulated too. And like I said, can be pretty complicated. I've taught and learned about biology for a very long time, and I don't fully understand the regulation of the cell cycle in terms of the molecular aspect. And honestly, this is very technical, and if we fully understood it, we'd have a lot easier ways to eliminate cancer, as we'll talk about later. And so there are definitely a lot of proteins involved. The only ones that you really have to know about for this class are called cyclins. It's a group or a family of similar proteins, and what happens is they alternate in concentration throughout the cycle, and by kind of fluctuating in concentration, they trigger different events to happen. Something that we're not going to talk about, I just want to point out in the pictures because that might be confusing to you, is we see these CDK, CDK, CDK. These are another type of proteins called kinases, and what they do is they bind to the cyclins and they kind of help them do their job. And so just know that there are several different types of proteins, and this is going to be where the regulation comes from. The cell is going to get these kind of messages or go-ahead signals from proteins and what's going on with them and their concentration. Now, another important part of cells and their activities is a process known as apoptosis. And this is known as programmed cell death. Okay, this is where a cell will essentially get a message to kind of commit cell suicide. It sounds really, really bad, but it's actually, for the most part, a good thing. Okay, apoptosis is a way for cells that are damaged to kind of eliminate themselves so that they don't get copied and cause problems. So if we got kind of these stop signals at the G1 checkpoint or at the G2 checkpoint, a lot of times there'll be this kind of chemical event that triggers the cell to kind of break itself down. And like it says here, it's normal for growth and development. When you were in the womb, you had a tail. As an embryo, you also had webbing between your fingers and toes, and now you don't. And so that's an example of cells getting told to die, and it kind of was just a normal part of that development. Now, this is also going to be important when we talk about cancer here in a little bit, is normal cells, typically, if they're damaged, will get the signal or get the message to go through apoptosis. Cancer cells, unfortunately, do not. And so apoptosis can help us prevent cancer by preventing the replication of these mutated or bad cells. And so here's kind of what's going on. So we start with a normal cell. Now this is going to be triggered through a chemical event. Through chemicals, the cell is going to be told to go through apoptosis. It's going to shrink up. You can see the membrane kind of shrivels up. The nucleus is destroyed and breaks down. The cells kind of starts to be broken up into different components. And then ultimately, they're going to be engulfed by these phagocytes that completely destroy and break them down. And so apoptosis can help us by getting rid of bad cells. It can help us with our immunity. If we have infected cells, it could keep those bad cells from multiplying. And so apoptosis really can be a good thing to kind of help maintain balance in our bodies. Now we also have some external regulation, and this could be through chemical signals that are secreted from other cells in the body. And so one example of these are growth factors. And it makes sense that growth factors play a role in the cell cycle because we grow by replicating our cells. And so growth factors are a type of hormone. They bind to the cell membrane, and essentially it's going to provide signals to the cell that they need to go through the cell cycle. And we also have some regulation when cells kind of touch each other. Cells have these that extracellular matrix that we talked about. It's got carbohydrates and proteins and different substances. It's got protein receptors in the cell membrane. And so when certain molecules either touch other cells or parts of a cell touches another cell, that can affect whether or not they will grow. And so we have two kind of regulation events. We have anchorage dependence, and this is where the cells have to be anchored to something solid. So if you just had like cells floating around in a liquid, they would not probably divide. They'd have to be attached to something like the bottom of a petri dish, or they'd have to be attached to another cell, another tissue, or some kind of membrane. And they won't really grow and divide unless they're anchored to something. And then another type of regulation is density-dependent inhibition. And this is what keeps cells from just getting crowded, okay? And so once they're anchored, they'll start to divide. Usually what they'll do is cells will kind of create a single layer, and then once they're touching other things, they will just kind of stop. A lot of times those growth factors can't get in there to tell them to keep replicating. And so once they filled up a space, they stop dividing. Now this is going to be a problem with cancer cells, is that cancer cells lack both of these kind of mechanisms. And so they'll just keep dividing, keep dividing, keep dividing. They just kind of ignore the anchorage and the density-dependent inhibition. So these are normal things that help keep cells from growing uncontrollably. Now we also have some gene regulation, okay? Now just to kind of be clear, these genes that I'm highlighting, proto-oncogenes and tumor suppressor genes, these are normal and these are good, okay? Now hopefully you remember that genes, they code for proteins. And so proto-oncogenes are going to be genes, pieces of DNA, that encode for proteins that tell the cell to divide, okay? They also prevent apoptosis. So proto-oncogenes promote the cell cycle. They encourage cell division, okay? They're going to lead to proteins that help promote the division of cells. Now, the opposite of proto-oncogenes are tumor suppressor genes. And these essentially do the opposite. They encode proteins that tell a cell to stop going through the cell cycle or to go through apoptosis. So you can think of the proto-oncogenes as like the gas pedal on the car. It's going to accelerate the cell cycle. But here's what can happen, is if it's mutated and we make too many of those proteins, or we don't follow that genetic instruction like we're supposed to, these normal proto-oncogenes can become what we call oncogenes. And you may have seen this prefix, this onco, before, like oncology, oncologist. That is a cancer prefix. And so proto-oncogenes are normal. They encourage the cell cycle. But if they become mutated, we call them oncogenes. So oncogenes are bad. Okay, these are the ones that can lead to cancer. And what happens is that when these cells become mutated, they essentially, it's like hitting the gas pedal and staying on the gas pedal. It's just going to keep going, keep going, keep going, keep going, keep going, and that's not necessarily what we need. Now, tumor suppressor genes are going to be like the brake pedal. They're going to be like the brakes in your car, and that they kind of slow down the cell cycle. So if we had a mutation in a tumor suppressor gene, they will not stop the cell cycle like they're supposed to. And so mutations in either the proto-oncogenes or the tumor suppressor genes are going to lead to uncontrolled cell growth. Okay. We also have some genes that, and there's more than one, but they're called DNA repair genes. And so, like, let's say in the G1 checkpoint, we noticed that the DNA was damaged. These produce proteins that help repair DNA. And so a lot of times the DNA will try to be fixed before the cell cycle continues. So if these guys aren't working properly and making the right proteins, the DNA may not be repaired properly. That will lead to mutations in other genes and could in turn affect the cell cycle. Okay. So this is just a few graphics. So here we have the proto-oncogenes like the gas. Okay. They're going to help encourage the cell cycle. The tumor suppressor genes are the brakes. They slow the cell cycle. So if you had mutations in either one, they're both going to lead to making too many cells, which is what we call cancer. And this is just the same thing over here, just kind of giving you a visual there for the gene regulation. All right, so what happens, we've already kind of mentioned it several times, but what happens if the cell cycle is not regulated like it needs to? The result is going to be cancer. Now, essentially, cancer is a very simple definition. It's unregulated cell growth. Cells lose the ability to regulate their division, and the process of developing cancer is known as carcinogenesis. So essentially, it's multi-stage. It's not like it just happens overnight. It typically begins with a mutation in either one of those proto-oncogenes or tumor suppressor genes. Now, what causes mutations? There's a variety of things. It could be an inherited thing, it could be a problem in DNA replication, or it could be caused by some kind of exposure. A carcinogen is just something that causes cancer. That could be something like a harmful substance like asbestos. It could be something like radiation. And so cancer is just when cells lose the ability to regulate their growth. And so what happens is these cells, instead of starting and stopping like they're supposed to, they just keep dividing, keep dividing, keep dividing, keep dividing. And what happens is they form tumors. So cancer leads to the proliferation of cells, which forms a tumor. Now, if you go to the doctor because you find a lump or a bump, there's typically one of two words that you're going to hear, okay? Now, the one that you want to hear is benign. And if you were to go to the doctor with a bump and they told you that it was benign, you would probably not come home and say, you have cancer, okay? Technically, cancer is just uncontrolled cell growth. But we say that we have cancer when a malignant tumor is discovered. And this is where it's invasive. Okay, so over here you can see that the benign tumor is contained. They're not really going to invade the surrounding tissues too badly. Now, they can still cause some problems, and normally what they'll do is they'll either go in and remove it or they'll just watch it. As long as it's not growing and invading nearby tissues, the benign tumor is not crazy harmful. Now, the problem is it could become a malignant tumor. So over time, this is why they watch them, because it could get to the point where it's invasive, it's spreading into nearby tissues, and it's causing harm. And so when we hear the word malignant, remember that mal or mal is bad in Spanish. Malignant are the ones that we say we have cancer, okay? Now, another problem with malignant tumors is the cells could break free because they're not really contained. The cells could break free and they could spread to other areas, and this is known as metastasis, or you may have heard the term metastasized, and this is where the cancer can spread to other locations. Now, you don't have to know a lot of details about this, but I wanted just to give you a little bit more because we are going to do a cancer project, and you guys are going to be researching a particular type of cancer. And so these are some of the words that you may come across, okay? There's carcinomas, sarcomas, leukemias, and lymphomas. And these are not the only ones. You can see there's some other ones here. But essentially, a lot of times the tumors or the cancers are named based on the types of tissues that they're invading. So carcinomas typically affect epithelial tissue like skin or the linings of your organs. Sarcomas typically affect connective tissues that are inside, that are making up your organs like bone, or it could be muscles. Leukemias form in the bone marrow, and these are going to be blood cancers where you have uncontrolled growth of blood cells. And then lymphomas is where it affects the lymph tissue, either in the lymph nodes or the other lymphatic tissue. So this is just a general type of tumors that you may come across, and really it's just because of what kind of tissue do you find the tumors. Now, causes of cancer, this can be pretty tricky because honestly, we don't know all of them, okay? And here's the thing, cancer stinks. There's not a single person in your classroom that's not been affected by cancer, or someone in their family, or a family friend. I mean, everyone deals with this in some way, either directly or indirectly. And one thing you have to understand is that we're living things made of trillions of cells, trillions. So the likelihood that 1 or 2 or 5 or 20 or hundreds cells are going to get mutated, the older you get, the more mutations you accumulate, which is why it's more common in older people. But all it takes is mutations in DNA, and cells lose the ability to regulate their growth. So we may not discover that cancer until it's harmful to us, but all it takes is uncontrolled cell growth. And so there's a lot of different things that could lead to those mutations. Now, obviously, if you expose yourself to things that we know are harmful chemicals, harmful substances, that is going to increase your chances of mutating that DNA in your cells. Sometimes viruses can affect the DNA in our cells, and that can lead to certain cancers. It could be toxic chemicals like asbestos or like, lead paint, things like that. Or it could be just basic old genetics. Some people are predisposed to certain cancers based on the genes that they inherited from their parents and their grandparents and so forth. And so we can't really put a pin on 1 or 2 specific causes, and different cancer types may have different triggers, but it's very common because we are organisms made of cells. Now, when it comes to the treatments, you're going to see some of these in your project. I just kind of wanted to give you a little overview here. A lot of times, if it's possible, they will go in and just surgically remove the tumors. That depends on how invasive it is and whether or not it's spread and how big the tumor is and what kind of tissue it's in. Commonly, you hear these terms, chemotherapy and radiation therapy. Chemo uses chemicals or drugs. And here's the deal. If you've known anyone that's gone through any type of cancer treatment, these make you feel pretty bad. Now, both of them are going to target cancer cells, and the ultimate goal is to harm and kill cancer cells. But what can happen is, in the process, you're exposing normal cells too. And so a lot of times you could have side effects where the normal cells are being damaged. But it's all in hopes that we can get rid of those cancer cells, and that's why you do see negative side effects. Immunotherapy is where they use substances to kind of boost the patient's own immune system and hopefully let the patient's immune system fight off those bad cells. There's hormone therapy where they use hormones to kind of stimulate or block cell growth. And then targeted therapy, this really plays in on those cell pathways. Remember, there's lots of different forms of regulation, so they have to figure out what regulation isn't working properly, but they will specifically target cancer cells and those molecular pathways. They may, increase proteins or decrease proteins, or they'll do something that will block a pathway that will hopefully prevent that cell cycle from continuing in those cancer cells. And that is going to wrap up our lesson on cell cycle regulation. And so you will see questions about this lesson when you take the test. However, we're not going to have a quiz over this particular set of notes because you're going to do a project, and you're going to get the details on that here shortly. You're going to have a couple of days in class to work on this project, and you're going to do some little short presentations of your cancer projects. And so instead of doing a quiz, you're going to review some of these concepts in a little bit different of a way. But I do want you to know that you will see this material on the test in the future. So don't forget about it. Maybe review it here or there as you're doing your cancer project. But we are going to do the project instead of a quiz, okay? That's all I have for you guys today. I hope you're doing well. If you have any questions, feel free to reach out, and I hope to see you soon.